Welcome to this explainer. We're jumping straight into the ultimate biological puzzle today, genetics. Have you ever wondered what actually makes you, well, uniquely you? Today, we are going to break down the exact principles of inheritance and variation. Let's start with a really simple question. Are you a 100% carbon copy of your parents? Obviously not, right? Even with the exact same parents, you and your siblings look completely different. So where does this variation actually come from? Why aren't we all just biological clones? Here is our roadmap for today. First, we'll go from DNA to chromosomes. Second, genes, alleles, and traits. Third, the difference between haploid and diploid. Fourth, genotype and phenotype. And finally, we'll wrap up with genetics and Mendel's famous experiments. All right, section 1, from DNA to chromosomes. Now, different organisms store their genetic blueprints in totally different ways. Take eukaryotic DNA. That's the type found in us, humans, as well as plants and animals. Our DNA is linear, it's kept super safe inside a nucleus, and it uses these specialized helper proteins called histones to pack it all tightly together. But then you have prokaryotic DNA, like what you'd find in bacteria. It's completely different. It's circular, it just floats freely in the cytoplasm, and it doesn't use those histone proteins at all. But think about our linear DNA for a second. It doesn't just float around loosely as a tangled mess. It packs itself incredibly efficiently. Chromatin is that condensed form of DNA wrapped around those histones. And it only forms those dense, familiar X-shaped structures that we call chromosomes when the cell is actually getting ready to divide during the M phase. It's basically nature's ultimate packing system. Moving right along to section 2, genes, alleles, and traits. So what actually is a gene? Simply put, it's the fundamental unit of inheritance, located right there on that DNA thread. It's a highly specific, targeted stretch of DNA that has a very real job to do. It controls a specific physical trait, things like your eye color, your hair texture, or how tall you're going to be. To make this instantly click, think about it like this. Just as a marker is designed specifically for writing, and a glass is crafted just for drinking, a gene isn't just some random piece of biological thread. It is a unit with a highly specific, defined function, which is to control a trait. You can think of every single gene as a tiny cellular worker with a distinct task. And that naturally brings us to our next concept, the allele. If a gene determines, say, eye color, the allele is the specific variation or flavor of that gene. It's simply a slightly modified version of the exact same gene, producing different variations of a trait. So it's literally the difference between inheriting the instruction for black eyes versus the instruction for blue eyes. Same gene, different allele. So where do all these genes actually live? Well, they have a very specific address called a locus. Think of a locus as the exact assigned parking spot on a chromosome, where a specific gene always sits. On a pair of matching or homologous chromosomes, the genes for the exact same trait will always park in the exact same parallel location. Okay, section 3, haploid versus diploid. Let's decode some vocabulary here. Haploid, which scientists represent with a lowercase n, means a cell has just one single set, or one copy of the genome. Think of a human sperm or egg cell, it's just a single genetic manual. Diploid, or 2n, means you have two full copies of the genome. Your skin cells, for example, are diploid. That means you have a primary manual and a backup copy, having inherited one from each of your parents. That brings us to section 4, genotype and phenotype. Now, the genotype is your invisible internal genetic makeup of alleles. It's the literal chemical code hidden inside your cells, which scientists write out as letters, like capital T or capital T lowercase t. The phenotype, on the flip side, is the physical result that you can actually see with your own eyes, like a physically tall pea plant. Essentially, the genotype is the blueprint and the phenotype is the actual building. Building on that blueprint idea, your genotype can be categorized as either homozygous or heterozygous. Homozygous means the two inherited alleles sitting at that locus are completely identical, like two capital T's or two lowercase t's. Heterozygous, though, means the organism inherited two completely different sets of instructions at that location, so a capital T sitting right alongside a lowercase t. This naturally leads us to how these traits battle for expression. A dominant trait is exactly what it sounds like, strong. It will express itself and be physically visible whether it's in a homozygous pair or a mixed heterozygous pair. It just overpowers the other instruction. A recessive trait, however, is a little bit shy.
It completely hides in the background and only expresses itself when it is in a pure homozygous condition, paired with an identical recessive buddy.
And finally, Section 5, Genetics and Mendel's Experiments. Stepping back in time to the 1850s, Gregor Mendel figured all these complex rules out without ever seeing a single chromosome or strand of DNA under a microscope. How? Well, his secret was the number 14. He meticulously started his experiments with exactly 14 pure, true breeding pea plant varieties. He painstakingly self-pollinated them over and over until he was completely certain his baseline data was perfectly clean and homozygous before he ever started mixing them together. With those 14 pure varieties ready to go, he looked at exactly 7 distinct characteristics of the pea plant. We're talking plant height, flower color, flower position, pod shape, pod color, seed shape, and seed color. And the truly brilliant part here? Each of these characters had 2 clear opposing traits, tall versus short, round versus wrinkled. There was absolutely no messy middle ground to confuse his data. Mendel kicked off this massive undertaking in 1856 and worked continuously until 1863. That is a full 7-year experiment. He formally published his brilliant work in 1865. But here is the tragic historical twist. The scientific community completely ignored it. It wasn't until the year 1900, decades later, that his monumental work was finally rediscovered by 3 separate scientists, at last giving him the credit as the true father of modern genetics. So, why did Mendel succeed where so many other brilliant scientists had completely failed? First off, he was the very first person to apply rigid mathematics and statistical logic to a biological problem. Real game changer. He also used a massive sample size, crossing thousands upon thousands of plants to give his data undeniable credibility. And finally, he continuously test-crossed his plants just to confirm his results weren't some random fluke. Let's break down his exact step-by-step logic, which we call his hybridization process. Step 1. He carefully selected those pure parents we talked about. Step 2. Artificial cross-pollination. He essentially played nature by deliberately dusting pollen from one specific plant to another. Step 3. He painstakingly observed and recorded the vast amounts of results. And Step 4. He used all that math to formulate the foundational laws of inheritance that scientists still use today. He perfectly isolated the variables to discover the truth. We've covered some incredible ground today. Mendel solved the deepest mysteries of inheritance and variation using nothing but pea plants, unbelievable patience, and math, without ever looking through a powerful microscope. It really leaves us with this final, provocative thought. In TVELV's modern era of rapid DNA sequencing, gene editing, and advanced biotech, what else is left for us to discover hidden inside our own cells? Keep wondering, keep asking questions, and I truly hope this explainer helped decode the beautiful blueprint of life for you today.